One day, as I was finishing up my delicious rotisserie chicken dinner, I thought to myself, how did all this food become what it was on my plate? Cells. They're the basic building block of life. I'm sure we've all learned about them in our biology class. Without them, we wouldn't exist, and Earth would just be another lifeless rock floating throughout space. So, how do you go from one cell and turn it into something that can feed an entire family? It's quite simple, actually. Cells need to eat too. Just like us, cells need nutrients to grow and survive. There are also some other growth requirements, such as the right pH level and temperature. I wanted to do an experiment to see what it would be like growing cells and analyzing them after. In this experiment, I will be culturing bacteria in different agar solutions and in different conditions to see which ones are best.
So here's what I'll be using. First off, I'll be using a total of 9 Petri dishes. I'll be using 3 different types of agar. So I'll be using nutrient agar, potato dextrose agar, and malt extract agar. I'm also going to need a scale for getting the correct measurement of agar, cotton swabs for collecting the bacteria, yogurt containers for making a warm environment, thermometer for making sure our temperature stays the same, and a microscope to see if I can analyze my findings. The first step in preparation is getting the correct amount of agar. So first I'm going to pre-weigh my bowl. Then, using the correct ratios, I'm going to add my agar. Since I'm going to be using 250 milliliters of water, I'm going to need about 5.3 grams of this nutrient agar. Next, I'm going to bring my 250 milliliters of water to a boil. After that, I'm going to add my water to the agar and stir until it is completely dissolved. In the absence of an autoclave, I'm going to use my microwave to mimic the sterilization process of the solution. I'm also going to repeat this in the previous 3 steps for each of the agars. After letting them cool for a few minutes, I'm going to put the agar solutions into Petri dishes, filling them about half to 2/3 and then swishing them around to cover the entire surface. I'm going to be doing a total of 9, 3 for each. Note that all of the utensils used were sterilized to avoid contamination. I am labeling each of my Petri dishes and I'm going to let them sit for an hour so it turns into a jelly-like substance. Alright, now onto bacteria collection time. I'm going to be swabbing the inside of my cheek as it is the most consistent source that I know of. Then I'm going to be streaking the bacteria across the surface, being careful not to tear the gel. The final step of preparation is creating the environments in which the bacteria will grow in. For the warmer environment, I'm cutting a hole in 2 yogurt containers so that I can shine light under and heat up the bottom without burning the dish. I'm going to use duct tape to keep the heat in, insulating it, and to keep the actual structure together. I already poked a hole in the top of the yogurt container so I could constantly measure the temperature. Also, I make sure to seal it up so that I can keep the heat in when I'm not measuring. Last but not least, the tin foil for that added insulation to make sure that it constantly stays at around 37 degrees Celsius. That's all the preparation done. One set will sit in a warm area, another in a room temperature area, and the last one in a cooler area. There we are. Here are the results of the 1 week cell growth, with number 1 being in the warmest temperature and number 3 in the coolest.
The results were definitely not what I expected. For starters, the constant heat led to a buildup of moisture, which is beneficial for cell growth. As you can see, nutrient 1 had a very spread out population. On the other hand, in the Petri dish labeled nutrient number 2, it was stored in room temperature, and it seemed to have the greatest buildup of bacteria in a small area. I think this is because, due to the lower heat, it was tougher for cells to separate from the pack and survive, just like hiking up the snowy peak of a mountain in a group. I was shocked that I could only see visible signs of bacteria growth in the nutrient 1 and 2 containers. This really put into perspective the importance of the nutrients that a cell gets. Although the agars were very similar, it was their differences that proved to be stronger. Cellular agriculture is an emerging field that uses the underlying concepts of cell growth to produce products that are identical to the real thing. Take a burger, for example. All you would need is one cell from a biopsy to produce meat that could feed an entire family. Pretty crazy. This could change the world. Cutting down on greenhouse gas emissions, and with enough scaling, it could be even cheaper and more widely available than traditional meats, giving this technology the power to attack two of the world's greatest problems: global warming and hunger. There are still a few major challenges in this field, one of which is finding a good long-term growth medium. Currently, the most popular one being used is fetal bovine serum, or FBS for short. It is derived from fetal cow blood, which raises some ethical concerns. On top of that, it's pretty costly, and it lacks that batch-to-batch consistency that is needed to scale up this industry. Developing a better media is key. I could tell from my experiment that even a small change in the nutritional contents will have a massive impact on the product. The move to an alternative will be challenging, as getting the right ingredients at a low cost is a very meticulous process. In the future, I can't wait to look back on this industry and see how much progress it's made as I take a bite into my nice, juicy burger that came from just a single cell.